How's it going, everybody? Today I want to go over this paper titled Rapid Biosensor Development Using Plant Hormone Receptors as Reprogrammable Scaffolds. This has to do with PYR1 biosensors, which is a huge emphasis of my PhD. It's about half of the PhD work that I did. I am somewhere lost in the middle of the authorship of this paper, so I contributed, very little to this paper, but my co-PIs are both on this paper, and I know a few of the other authors involved, and I built a lot of my PhD off of this work. It's also a very high impact paper, so it's in the journal Nature Biotechnology, which isn't just Nature, but it's maybe one step below that, which is still, really big impact. And so maybe a little bit for my future self, but also anybody else from my lab or anybody else interested in the sort of in-depth and the lore of the PYR1 biosensors. I think this video will be really helpful for you. So stay tuned. I know it's a long video, but I think it will be interesting if you're interested at all in these PYR1 biosensors. So let's dig into it. All right, so I already read the title. The first author here, the first listed author is Jesus Beltran. He is now a professor at the University of Delaware, but he was a postdoc in the Sean Cutler group, and he was a mentor for me. He showed me how to work with these PYR1 biosensors. So shout out to Jesus, really, really sweet guy. If you have a chance to work for him, you definitely should. But what isn't super clear here in this publication is that these four first listed authors are all co-first authors, which is kind of rare for a paper. But it just sort of indicates that this was a big collaborative paper between a lot of people. Shuang Wei was also a mentor for me, and that worked directly in my lab. I'm in the Ian Wielden lab. And she's now up at, she's working in the Bay Area. I forget which company, but she's in industry now. So she's also a co-first author. And I know a few other people on this paper. Sang-Yol Park is a staff scientist in the Sean Cutler group, and him and Sean both sort of developed the PYR1 biosensor technology themselves in their lab. So they sort of kickstarted this whole network of research. So really high impact there as well. If you go to his Google Scholar, you'll find a lot of papers on PYR1 biosensors if you're interested in that. So I'll go through the abstract pretty slowly. It'll be a big chunk of the video because it's very condensed. And then I'll go through the sort of the introduction a little bit as well, tiny bit on the results and discussion, and then mainly talk my way through these figures because the figures are really what's going to tell the story here, right? If you want to go and read the paper in detail, you can, but I'm not going to read it out loud to you. So we'll start with the abstract here first. So there's several other papers you can go read before this one, and there's also a few patents around this technology. The beginning of the abstract, a general method to generate biosensors. So we're talking about biosensors for user-defined molecules. So this is sort of claiming like, we can go and find a chemical that we want a biosensor for and then use PYR1 to find biosensors for it. So user-defined, that's what we mean by user-defined here. Could provide detection tools. So we're talking about tools now too for a wide range of biological applications. So we might call this flexibility here, between chemicals, and then portability between applications. So this is big for biosensors. This is really big for biosensors. So here we describe an approach for rapid engineering of biosensors using PYR1. So now we're finally talking about PYR1 specifically, which is pyrrobactin resistance 1. It's naturally a hormone receptor in plants for abscisic acid, or ABA. We may see that abbreviation later. And the binding pocket of that protein is malleable. So if you want to see sort of a more broad overview of PYR1, I have another video on my channel that's only 5 to 10 minutes long. But I talk about the binding pocket there. But basically, PYR1 is a protein with a binding pocket for that, naturally for that ABA compound that fits in there. And this is basically saying that we can change that binding pocket to detect new ligands or new chemicals, right? So malleable ligand binding pocket, and a requirement for ligand-induced heterodimerization, which facilitates the construction of sense-response functions. Okay, so the heterodimerization will come up again later, and we'll talk about that. Sense-response functions, basically saying that, this sensor will detect something and then give us an output, which is, a sensor is kind of useless if you don't have an output. So we'll show that in this paper as well. We applied this platform to evolve 21 sensors. That's not very many. These are just the ones that were published. And these have nanomolar to micromolar sensitivity. So for some context, there's other biosensor technologies out there. Most of them only get to micromolar sensitivities. So getting to nanomolar sensitivities is a big deal. And the chemicals that were for a range of molecules, a range of small molecules, small in this case, is basically meaning not proteins. So anything under like 500 grams per mole is what we consider small. So including, what chemicals are we testing for? Including structurally diverse, so looking at very different chemicals, natural and synthetic cannabinoids, cool, organophosphates, which are a class of pesticides, also cool. And we have biosensors for lots of other chemicals now too, but this paper is just looking at these cannabinoids and organophosphates. Also in this paper is some X-ray crystallography, which is basically a way to look at the structure of the protein. And through that structural analysis, we found a mechanistic basis. So basically how the mechanism works for the ligand binding in a mutated protein as well. So we look at the crystal structure of the wild type protein that binds ABA, as well as one of the mutant proteins that binds to a cannabinoid. So PYR1 derived receptors are readily ported to this portability, again, to various ligand responsive outputs, including ELISA-like assays, which is an in vitro assay, so outside of the cell. This is a very commonly used lab test for small molecules or other chemicals. And so being able to move PYR1 into an ELISA-like assay is significant. We also showed that PYR1 can be used in a split luciferase assay, so luminescence assay, and I'll talk about that more later, as well as transcriptional circuits in bacteria and yeast, which is cool. And all of these have picomolar to nanomolar sensitivity. So you have molar, and 1,000 times less than that is micromolar, 1,000 times less than that is nanomolar, 1,000 times less than that is picomolar. So picomolar sensitivity is pretty significant. Okay, and then sort of a conclusion sentence here. PYR1 provides a scaffold to rapidly evolve new biosensors for diverse sense response applications. Okay, that makes sense now. Okay. Getting into the introduction here, this first chunk is basically laying out the problem and laying out what's already been done in the field before. So very common for a beginning of an introduction, saying that we need specific and portable biosensors for biotechnology applications. Great. So there's a need. Wonderful. There's competitors, so a number of protein scaffolds talking about bacterial allosteric transcription factors. Usually with these bacterial allosteric transcription factors, though, they only work in one organism, or they only work in bacteria, right? And so being able to move these biosensors between species and into in vitro assays is significant. I don't know too much about these G protein coupled receptors, but I'm sure they have limitations. And there's some computational designing of proteins that we can do as well, and they cite other methods. So yeah, each of these technologies are great, but there's limited scope of what chemicals we can detect with these, and limited applications of these existing biosensors. So we need new biosensors for biotechnology, right? So this is setting up the motivation and sort of the background. So now we're talking about chemically induced dimerization, or CID. So this is how the PYR1 biosensor platform works, and the way that it works is there's the PYR1 protein that detects the chemical, and then another protein called HAB1 or another protein phosphatase, which we'll look at in a second, comes and binds to PYR1 only when that chemical is present, right? So that interaction we can sort of hack for other applications. And it has a few advantages, one of them being that having, if you just have one receptor and no other partner chemical, if you don't have that dimerization going on, then that chemical can easily come out of the binding pocket. But if you have that other protein coming and docking, it sort of locks in that ligand, which increases the sensitivity of the sensing system. On top of this, there are other CID modules out there, but usually you have to mutate both proteins. But in the PYR1 system, you only have to mutate one of the proteins. And what this means is, the ligand binds only in PYR1 and not in HAB1 at all. It doesn't interact directly with HAB1. So if we want to change what chemical we detect, we only have to mutate one protein, and the other one can just stay the same the whole time, which makes it, I don't know if exponentially is the right word, but it feels like exponentially easier to engineer new sensors. All right, so two proteins form a stable heterodimer only in the presence of the small molecule. Great, because they rely on single protein-protein interaction, right? Localization, CID, ligand binding is shared between partners. So this is talking about other CID systems. Usually ligand binding or chemical binding is shared between both of the proteins, which necessitates redesign of the entire interface. So redesigning both proteins. And PYR1, the PYR1 system is different. So the ABA sensing system functions naturally occurring CID mechanism. Ligand recognition by PYR1 leads to the formation of a PYR1 ligand protein phosphatase 2 complex that inhibits phosphatase activity. I'll talk about that in a second. What I wanted to get at, though, for right now, though, is this system is unique because ligand recognition occurs exclusively within PYR1. Significant. So this protein phosphatase 2 or PP2C, HAB1 is naturally in the plant. PYR1 is just a receptor, and HAB1 is a protein phosphatase. So without ABA present, the protein phosphatase can go around removing phosphate groups from proteins, causing downstream cascades, right? But if PYR1 has a ligand in its binding pocket, then it can bind to HAB1 and prevent, so it inhibits that protein phosphatase activity. And we'll see this in one of the figures in a minute, and we'll talk about it more. So that's sort of how the natural mechanism happens. All right, so in addition, the phosphatase acts like a co-receptor, and having HAB1 sort of cover up that binding pocket boosts the apparent affinity up to 100-fold, like I mentioned earlier. So we can get micromolar PYR1 binders. All right, our data shows that high density mutagenesis, so we're going to be mutating that PYR1 binding pocket, rapid development of sense response actuators portable to in vitro and in vivo control systems. Really, really cool stuff. So in the beginning of this results section, we're talking about generating our first mutant library. So if we mutate that binding pocket of PYR1, we're able to create new sensors for different chemicals. So, if we put a really big amino acid in there, then maybe abscisic acid can no longer fit in the binding pocket, but another chemical can fit. And we'll see what that looks like. But the two things to remember from this first paragraph are we made a library of about 36,000 mutants. So that's how many mutants we're screening, and we call this library the DSM library, or double site mutagenesis library, meaning we're just changing. So a single site nicking mutagenesis will change an individual amino acid, right? So a double site mutagenesis library will change up to two amino acids within that binding pocket. So you'll see that in a second. So this first panel A of figure 1 is in part showing the crystal structure of PYR1 and HAB1 and abscisic acid bound. So abscisic acid is in yellow here. Making sure I'm still recording. So abscisic acid is in yellow here. The blue amino acids are the amino acids that Sean Cutler chose to mutate that are within that binding pocket that actually interact with abscisic acid. And in the green here, here's HAB1 coming in and forming this salt bridge with PYR1. Very, very cool, right? So by mutating these blue amino acids, we can change which chemicals our sensor will detect. And we didn't change them to all possible amino acids, or Sean didn't choose to change them to all possible amino acids because the library would just be tens of millions or hundreds of millions of mutants big, which is not reasonable to screen. So he said, okay, here's a phenylalanine here at the 159 position in PYR1. What amino acids could we possibly change this to mold the binding pocket? Do we want to change it to a more a positively charged or negatively charged amino acid, or a much smaller amino acid, or what? And so there were some decisions made about which amino acids we want to mutate to. Okay, panel B is showing sort of a cartoon of how this mutant library works. So basically we have this set of, so we're mutating the DNA sequence of PYR1 to change the binding pocket of the protein. We can mutate just, one position on the protein, or we can mutate two positions at the same time, and we're pooling this set of mutants together to make the DSM library. And when we do this screening, we're finding, hopefully finding a biosensor for a new chemical or a ligand of interest, right? And it says Y2H screening right here. This is yeast 2 hybrid screening. This technology is like at least 2 decades old, I'm pretty sure. And the idea here is that the yeast have all of the machinery that we want in order to do our screening. So the yeast is able to take a DNA sequence, convert it to a protein that the chemical that we want to test for is able to come into the cell to some degree. That interaction can happen. We can have transcriptional circuits in the yeast. And then at the end of the screen, we can pick a yeast cell and sequence the DNA to find out which sensor or which sensor sequence is actually in the yeast. So we have to do it in yeast. They have all of the machinery that we need. So the way that we do these screens is that we'll have a transcriptional circuit that's activated by the binding of PYR1 and HAB1. So PYR1 and HAB1 come together. I'll show the circuit in detail a little bit later, but PYR1 and HAB1 come together, activate transcription of a gene, or in the case of the yeast 2 hybrid screening, 2 genes. One of them is URA3, which encodes for production of the amino acid uracil. So this yeast has a deletion of the normal URA3 gene, so it can't make URA3 on it, or it can't make uracil on its own. It needs uracil in the media, or it needs this URA3 gene to be transcribed. And the only way that that URA3 gene is going to be transcribed is if there is a biosensor in the cell that detects the chemical, binds to HAB1, and then transcribes the URA3 gene. So basically we put like millions of cells on a petri dish with our chemical. All of the cells will die except for the ones that carry a biosensor that detect our chemical. So this is how the screening is set up. And then to validate that sensor is real, we can do these beta-galactosidase stains. So the way that this works is, the chemical is detected, PYR1 and HAB1 come together, and it transcribes beta-galactosidase as well, which through some mechanism makes the cell turn blue. So we basically stain the sort of a spot of the yeast cells, and if it turns blue, then that's how we know that the sensor is being activated, right? So that's what panel C is showing here. So this is I believe this is a synthetic cannabinoid, and we found this set of 5 hits from the DSM library and decided to do DNA shuffle experiment, which is exactly like it sounds. It's like shuffling a deck of cards. So we're taking all of the mutations and mixing them all up, making a new library, and then screening for the same chemical to find a higher affinity or lower sensitivity sensor. So this is going from some of these sensors are around the 1,000 nanomolar range, so 10 to the 3 is 1,000, so 1,000 nanomolar range. And then after the shuffling, we found 100 nanomolar sensitivity sensor here. So basically, we make this library that has a bunch of almost random mutations, not sort of directed mutations towards any one chemical. We screen a bunch of chemicals and hopefully we find a few hits, and then we can shuffle these DNA sequences together to get a higher affinity or higher specificity sensor at the end. So that's sort of the pipeline that's going on here. This section is talking about what these mutations are, what chemicals we're screening for, and why. And so figure 2 here is just looking at cannabinoids now, both natural and synthetic cannabinoids. So the natural cannabinoids are like CBDA and delta-9 THC, and then the rest are all synthetic cannabinoids. Still recording, great. So the way that you read this, you read across the row, right? So this is a CBDA sensor at position 59, which is normally a lysine. So normally it's K59, has been mutated to an arginine, or the amino acid single letter symbol is R. So this is a K59R mutation, and then I forget what's that. So yeah, so then F159, so 159 is normally a phenylalanine. It can be changed to a valine to be sensitive to this particular synthetic cannabinoid. So this is showing, after a DNA shuffle experiment, higher affinity sensors here. And then this column is showing the limit of detection of the most sensitive sensor in nanomolar. So some of these go down to 10 nanomolar, which is cool. Some of them stayed at, in this case, what is this, 100,000 nanomolar, so 100 micromolar for this sensor. So it really depends on the chemical, what kind of sensitivities we can get. And then this heat map on the right is a chemical similarity chart. So it actually doesn't have a color key here, but the more blue, the more similar these chemicals are. So this is basically just showing we're looking at structurally diverse chemicals and we're still getting biosensors for these different chemicals. Panel B is showing, some more beta-galactosidase staining for different biosensors. So this one is for CBDA, sensitive down to 250 nanomolar. This is the first sensor in panel A, and then some other biosensors as well. So like this one, this WIN, I think it's WIN55 is the chemical name, goes down to 10 nanomolar. So that's cool. Panel C, D, and E are showing some crystal structures in their binding and whatever. Cool, fine. So talking more about sensitivities and what these sensors actually do and what it means. Figure 3 is showing the in vitro assays that I was talking about earlier. So this is a big part. So that first section was maybe showing more of that flexibility of the platform, that we can detect a wide range of chemicals and make them pretty, make the biosensors pretty specific. And then figure 3 is addressing that portability claim of we can take the biosensing system, into different organisms, but also outside of organisms entirely, and develop these lab assays for different chemicals. So now we're looking at cannabinoids still, so natural and synthetic cannabinoids. So this panel A is showing the sort of wild type mechanism of PYR1, and so the y-axis is %HAB1 inhibition. Remember, HAB1 is the protein phosphatase. When PYR1 binds, it inhibits the protein phosphatase activity. So we use a chemical here that when the phosphate group is removed, it changes color, and so we're able to develop this titration curve in that assay. And then we can also move this system into yeast for a GFP assay. So now we're looking at fluorescence. So GFP is green fluorescent protein. It fluoresces green. And so that assay works in yeast. And then another assay in yeast is a split luciferase assay, where luciferase is a protein that luminesces. It doesn't fluoresce, it luminesces, so it generates its own light. And so we're looking at luminescence on the y-axis now, and that split luciferase with the same sensor works as well. And for all of these, we're getting on the order of about 50 nanomolar sensitivity, right? So that's really cool. And then finally, we have this ELISA-like assay. I don't remember the specifics of how the ELISA-like assay works, but we're using the ELISA-like assay here. This was a collaborator group. I'm pretty sure the Tim Whitehead group did this experiment. And so we're looking at absorbance at 450 nanometers for whatever reason. Again, I don't remember how the ELISA assay works. But the ELISA-like assay can be used for drug screening panels or looking at chemicals in human liquids. So we have simulant, saliva, urine, and serum, as well as a saline solution. And in the ELISA-like assay, we actually get much higher sensitivity, about 2 orders of magnitude higher sensitivity, down to picomolar levels of sensitivity, which is really, really fantastic for future applications of this biosensor. And then finally, in panel E, this is an ELISA-like assay again for using this Win55 sensor, showing cross-reactivity. So we can look at all of these other cannabinoids, and the only one that this biosensor detects is Win55. So that's great. We've got good specificity. I remember there was, when this paper was getting published, there were some reviewers that were skeptical of the selectivity of these biosensors. So they were basically saying like, oh yeah, sure, you have sensors for these other molecules, but you're just opening the binding pocket so much that anything can fall into that binding pocket, which is a valid criticism, right? And I remember Sean and Ian being pretty frustrated having to generate all of this cross-reactivity data, sort of shoving it in the face of the reviewers like, look, this isn't just about expanding, opening up the binding pocket. This is about creating a completely new biosensor that's specific to new chemicals. So that was needed, but understandable frustration as well. And then so finally, we're looking at organophosphates now. Panel A is really similar to the panel A of figure 2. Panel B is similar to figure 2 as well, looking at a couple of the organophosphates. And then panel C, we're looking at a luminescence assay. So this is a bit of a cross-reactivity bit here as well. So this is looking at a sensor for the organophosphate called diazinon and the organophosphate called pyrimidphos. And so this is a luminescence data, so this is the split luciferase assay. And then panel D, we're looking at the sort of wild type function of PYR1 and HAB1, because we're looking at percent HAB1 inhibition on the y-axis. And then we're also looking at the beta-galactosidase spot plate staining, looking at cross-reactivity here. You can see the diazinon sensor is specific to diazinon, and the pyrimidphos sensor is specific to pyrimidphos. But in the in this HAB1 inhibition assay, there is a little bit of cross-reactivity going on, which is what we expect, especially at really high concentrations of the chemical. And then finally, this is another assay showing cross-reactivity with other organophosphates, but we're looking at fluorescence now. So this is GFP reporting, and you can see that our biosensors for diazinon and pyrimidphos are specific, but there is some cross-reactivity. Of course, there's going to be some. If you add enough chemical, it's probably going to bind in similar sensors, right? Okay, so that's all of the main text, and then we have some methods here. I want to go to the supplementary information just briefly and go over that pretty quickly. There isn't too much in here to go over. So this is a huge cross-reactivity assay with wild-type PYR1 to show that none of the chemicals interact with wild-type PYR1. So we actually are mutating the PYR1 binding pocket to new chemicals, right? So that's important data to show. Some more cross-reactivity stuff. So this is for the CBDA biosensor. This is for one of the synthetic cannabinoid biosensors. The biosensor doesn't cross-react with anything else. Again, driving home that point. This is some of the GFP assays and like how we did the flow cytometry analysis of that. I think Schwang did that, I believe. This is some more crystal structure stuff, looking how things bind. some more cross-reactivity stuff, but this time with the ELISA-like assay. And then this is more ELISA-like assay stuff, looking at, I believe, cross-reactivity as well. Okay, so anyway, that's basically it. I think this is a really cool paper. I'm a little biased because I'm one of the authors on it, but I think these biosensors are super, super cool and powerful tools, and if you have an opportunity to work with them, I definitely say go for it. I really want to see these move into a test strip application, so you can have like a 10-cent test strip for, you know, any cannabinoid, specific cannabinoids, or a synthetic opioid like fentanyl or something like that, or, you know, something like testosterone. That would be a super powerful test strip candidate as well. And as far as I understand, the PYR1 biosensors have not been used in mammalian cells at all, and I don't see any reason why they couldn't be used. So I think that could be really useful as well for, I'm not a mammalian cell person, I work with yeast, but I think that could be really cool. Oh, and I also wanted to mention the only contribution I really made to this paper is one or two of the CBDA biosensors that I discovered made it into this paper. I found a bunch of other cannabinoid biosensors, a bunch of THC and CBDA and other cannabinoids like CBDVA. There's a bunch of cannabinoids, and I found biosensors for a lot of them. So anyway, I think this paper is cool. Thank you so much for coming on this journey with me. If you have questions, please put them in the comments below. I'll try to get to them as quickly as possible. You can look at my Google Scholar if you're interested in that kind of stuff, to see similar papers. So if you want to read more, you can look at my Google Scholar. Nicholas R. Robertson is my sort of Google Scholar name. You can look at the Sean Cutler group for more biosensor stuff, or the Tim Whitehead group, or Ian Wheeldon's group, or Samuel Park did a lot of the pioneering PYR1 biosensor development. That one is good for more historical PYR1 biosensor stuff. So anyway, don't forget to subscribe to the channel to see more content like this. If you like this kind of content, comment below. That would encourage me to do this more. Make sure to like the video. Thanks for watching. Bye.